Welcome to the course, everybody. Today we'll do like, instead of a class, we'll do it like a demonstration of what a Mac can do for bioinformatics. Usually people are scared of bioinformatics, but there's nothing to be scared of. It can even run on a Mac, it's a smiling Mac. Okay, you might ask why you want to use a Mac. Because Mac OS 10 is Unix. Oh, there's one more. Couple of, We're just on a second slide, so it's okay. So Nicole might ask, why do we want to use a Mac for bioinformatics while you can use PC or Linux? Because Mac OS is Unix. Mac OS 10 is Unix, which means you can use a lot of Unix programs out there that are available for bioinformatics, which are essentially real industrial strength bioinformatic programs. And because of that, you can use a single computer to do both day-to-day. Hi. And there's a handout over there. If you want. So you can do both day-to-day applications like running Microsoft Office, email, web browsing. At the same time, you can also use a Mac to develop your bioinformatic workflow. And also, by using a Mac, you can integrate graphical user interface programs with graphical user interface and command line operated programs. And they can be combined together seamlessly just by alternating between the windows. And also, my largest argument is just elegant. Hi.
Hi.
That's a handout if you want. Okay, what do you do on a Mac? This is our procedures that we follow in our lab. So essentially, you start your raw material is DNA sequence. So DNA sequences can be from like ESTs or shotgun genomic DNA assembly or next generation sequencing sequencing reads. And you can process these several nucleic acid files on a cluster, which is a bigger computer, which have lots of memory and use a lot of processing power that more than any one computer can do. So what a Mac enables you to do is to connect from your desktop directly to this cluster system by using secure shell login, which when you do real bioinformatics, and then you will start using the terminal programs, and then you will know what SSH means. But for now, what I like to say is Mac can come in between you and the cluster programs. So you can do it also on PC, of course, but it's a little bit cumbersome, and you have to go through a little bit more extra steps to transfer one file from your PC to the cluster. But if you use Mac, you can drag and drop, essentially. Okay, what would you get from the raw DNA sequence? You'll get translation of your open reading frames and prediction of gene models. What I mean, gene models are like exon and introns for a hypothetical gene that you don't know how it will look like, but you are just guessing from your previous experience or from an EST sequence database. And the organization of these exons and introns are predicted by programs, and then they produce these gene models for you to look at and then see to assess what you will get. And also you get more assemblies using Velvet and Sasaki and VCake and so on. And then what you end up with is another off. So that's what you want to look at, ultimately the amino acid sequences. And then you derive sort of what could be its function, usually looking at this amino acid sequence rather than nucleic acid. Another thing you can do on the cluster is do alignments of short reads, very short reads, which are, in the past it used to be 36, now it's 72. And these days you can also have pair ends reads, depending on your insert size. It could vary from 156 to 300 if you want to assemble them. So once you have those things, and then you can align them to your reference genome. So they are particularly useful if you have a genome sequence from one organism, and then you want to look at very closely related organism, and then figure out what the differences are between the two organisms. So to do that, you need either Mac or BWA alignment programs, which will take small reads and then align them to your genome, and then report the positions and coverage of how much is being lined up in that particular position. And then the information that you can get out of SNPs and coverage of the reads, and you can derive some sort of idea as to how many gene copies you have in that particular organism, which are different from the reference organism. Okay, so you can do all this on the cluster, but Mac can come in and then connect you to the cluster. You can download, upload sequences quite easily. So that's another reason that you use Mac. And another follow-on workflow that we use Mac for is this. So amino acid sequences, as you see from the previous slide, derived from different methods. What you want to know is, what do they do? Maybe you are interested in effectors. Maybe you are interested in resistant gene-like proteins or signaling proteins. You might be interested in some other transport systems. So you can use several methods to guess the function. So first line of attack is to do BLAST, to see homology search against a database which is annotated with function, and then you see what that protein might be doing. Or you can do hidden Markov model search using Hammer, which can take protein motifs or protein HMM models from PFAM database, and then look across your protein that you are interested in, and then see what the match is, and report all the matching PFAM domains or HMM models. And also you can look at whether the protein is secreted or not by running signal P, and then doing BLAST against protease database that I'm particularly interested in, and then you can figure out whether a protein is likely to be a protease or not. And also some people will be interested in doing clustering of these amino acid sequences. By clustering, you are creating gene families. So gene families can be assumed, if the sequences are closely related together, can be assumed that they are derived from a single ancestor. And then by looking at the mutations within the sequence or changes between the sequences, and then you can guess the history of that particular gene, or the history or evolution of that gene. And also you can look at phylogenetic analysis once you get the amino acid sequences from different organisms, and then you can derive how related they are to each other. Or simply, if you know a motif that you want to search, for example, our lab is interested in RxLR proteins, so you look for RxLR, presence of RxLR in every amino acid sequence that we can find. And then what you will get is you will get annotated sequences, or you can have changes in the protein and evolutionary history of the protein, and then you have candidate effectors. So that's the bioinformatic flow that we do in our lab. So that's what I want to present today. Okay, there are several programs that we use a lot in our lab. First is called BBEdit. It's a simple text editor, but it's got these three strengths that is very useful for us. One is it can open really big files, like 300, 400 megabases or megabytes with millions of sequences in it. And it can search across multiple files using a search and replace function called grep. And grep uses regular expressions. I'll come to that later. That is part of a bioinformatic jargon. Regular expression in simple term is just using the wildcards, like star or little dot, something like that. So, for example, if you want to translate this text line into a regular expression, you'll have something like this. So that's just a code to find this text. So BBEdit can do that. So for example, if I want to find the protein, this homolog on this ID protein, I could use this thing to look at what matches in this parenthesis, and then I can save this in a variable, and then I can print it out quite easily in BBEdit. So that's why we use this BBEdit program, and it's a simple text editor, yet it is very powerful. And I'll make a demonstration later on in the course. Another thing that we do is we do a lot of sequencing of our clones. And a lot of people just do manually. They have the sequences, do alignment, and then see what they are. But in our lab, we have a systematic approach to sequencing projects. And for that, we use Phred and Sequencher. Phred looks at trace files, and then it produces a quality score from an algorithm that I don't know much about. But that quality score is reliable, and you can assign a quality score to a base, and then you want to call it base or not, depending on that quality score. And Sequencher can read those quality scores and then import them into a project, and then make an automatic alignment, and then report back whether any differences from the reference or there is any polymorphism within the sequence reads. And Sequencher can also do low quality trimming, vector trimming, and contig assembly. So that's what we use in the lab. And then you can edit all the ambiguous bases and find SNPs and align with reference sequences and etc. So, and Phred not usually, but the only thing that way that we can run Phred is by command line these days. And it's quite simple. You open up a terminal and then type in this sentence, and then you'll get your quality score. But I'll come to that. I'll come back to that later. And this is a screenshot of what you can get from Sequencher. So these little arrows represent your sequence read from ABI machine, and then it's being assembled together and forming a contig. And what neat things about it is, you can see that they have forward or reverse directions, and it has 3 reading frames that you can look at. In this particular case, the first reading frame is deprived of stop codons. I know that because these little P's on the sequence that's line is start codons, but these red lines are all stop codons. So for the first frame, so there's a free of stop codons until the end. So that's why I know that it's the correct frame. And then once you get the correct frame, then you can look at where the snips are. What else do I want to tell? Okay, so this is just an overview of what a sequencer look like once you have assembled. And there's another program that I use quite often. Is MacVector. Some of you might have used Vector NTI. Anybody use Vector NTI? You use Vector NTI? Okay, Vector NTI is the program to draw vectors and make constructs and do gateway cloning, in silico gateway clonings and stuff. And MacVector does the same thing. Well, with Vector NTI, they stopped making a program for Mac. So current Mac OS cannot run those Vector NTI anymore. So we use MacVector a lot. This is pretty simple, really. You just copy and paste your sequence in, and then highlight the sequence, and then make it into a feature, and then you can assign different figures for that particular sequence based on what they are. For example, this one, I have a BLD3 sequence. So this is a gene sequence, and that's off with no introns. And I've designed some primers to amplify promoter region, and then a little bit of terminator region. So, and then you can diagrammatically represent that event. So MacVector is another program that we use a lot. And that's sort of figure that you can produce in MacVector. That's a publication quality pictures, which other programs like Apes or something like that cannot produce it that well. But MacVector can do that. The only problem with MacVector is it doesn't have a database. So you have to make your own database, just saving files in one folder. And otherwise, it's pretty good. Okay, another program that you will probably need if you do bioinformatics a lot is DNAStrider. It is a very fast and versatile sequence analysis program. You can paste in your sequence, and then you can translate any region you want. 6 frames, 3 frames, 1 frame, forward, reverse. And you can do restriction enzyme digestion, and then you can compare 2 sequences if you want. And also, I use that program to do off finding. If you have a stretch of DNA sequence, you paste it in, and you keep clicking to find offs, several offs in the same sequence. I'll come to that later. And here's another useful program called Finch TV. It's very simple, really. It just reads in your trace files and show it diagrammatically along with the quality scores on the top. And what program what this program can do is it can print the metadata for that particular sequence file. So metadata is like which machine it was run, where the file is, and then what die it used, what die format that it's used when it was run, so that Phred can use this data to make a quality score calling. So to configure Phred program, you need to have this file at this name to put it in his configuration file, so that Phred knows how to call quality sequence, a score for the quality score for the sequence. So I use this program to find out what that file is. And it's also very easy to look at the quality of the sequence reads. Okay, another program that you will need is called Enzymex. And it's simply it's a program that spits out restriction sites and what the buffers that you need to use, and what would you do if you want to do double digest. You don't need to look up the chart and stuff like that. So you just type in 2 enzymes and then it would suggest a buffer to use. And also you can ask questions like that. What other enzymes?
Sorry for my ignorance. What's a double digest?
Sorry?
What's a double digest?
Oh, double digest is you use 2 restriction enzymes in one reaction.
Okay.
So sometimes those 2 enzymes need different conditions. One enzyme might need higher salt, one might need lower salt. So you need a compromise for the buffer. So
does this have sort of like restriction enzyme cutting sites and stuff like that? Is that basically what it is?
yes,
So it'll tell you if you get an enzyme, it'll tell you whereabouts in your sequence that will cut.
this will also tell you, yes.
Okay.
Yeah, if you paste in a sequence,
Yeah.
and if you choose the enzyme and choose the enzymes, and then you will get fragments. I'll show that once.
Is that what's
Demonstration
missed by that first step? What is the recognition side of pack?
No, this is just a restriction enzyme,
Yeah.
and you want to know what sequence that restriction enzyme would cut.
Okay. Okay, just one sequence.
Yeah.
Okay.
So it would tell you the sequence recognition sequence.
Right. Okay.
So I'll also be showing how that program works. And my favorite, amplify program. And that's very useful to quickly check your PCR primers, and then calculate amplicon sites and see what you will get from your primers just from that template that you would use. For example, I have designed some primers here to amplify SGT1, and then it shows you where the primers will anneal, and then shows you what are the predominant amplicons that you will get if you use all primers all together. And I'll show that as well in the next. Okay, after all that analysis and workflow that you finished, what you do with your data, you have to store them in a readily accessible form. We used FileMaker Pro, which is a database program that you can use and you can configure really easily. Just drag and drop. If you can, make boxes and tick marks on a PowerPoint kind of a presentation, you can use this program. So we use that program to store our annotation data, and then we have all the expression data and Illumina sequence alignments. We use this FileMaker Pro to store all those things. And also, you can use this database to search or to mine what you want to see. For example, we have 18,000 sequences from phytophthora infestans. So you want to know which ones already being sequenced. You click on a sequence box, and then you do search. It brings up all the ones that have been sequenced. That's for example. Okay, those are the programs that, the essential programs that we use all the time. So I would like to go into a little bit. I would like to go a little bit into Unix side of bioinformatics on the Mac. Before I do, I would like to mention that all our sequences are based on this format, which is the FASTA format. And this is a very simple format to represent your sequence. It doesn't matter whether it is a protein sequence or DNA sequence, the same format. So for this, for the subject and identification lines, you have a greater than sign in front, and then one line. So anything between greater than sign and the first space is the ID of this sequence. And the rest, from the first space character until the end of the line, is called description line. So this is the ID. This is the ID, and this is the description. And the rest that follows that line is assumed to be sequence. So you have to put the sequence after that. And you can have multiple lines of sequence after that. And also, you can bunch up several sequences together in a FASTA file. So that becomes a FASTA file with multiple sequences. Okay, yeah, so that sequence doesn't have any differentiation between nucleotide sequence, nucleotide file, and protein file. So we usually have a what do you call that? So that we know which file they are. We have a little extension for nucleotide files. We use FAN for FASTA nucleotide, and for protein files, you use FAA, which means FASTA file amino acids. And if you know FASTA file, you can do bioinformatics. And another thing that you will probably need to know if you want to do bioinformatics on Mac is the command line terminal. And that command line terminal is the window to the Unix world. Once you know how to use this command line terminal, and then you are open to a lot of software programs out there that can process these DNA or RNA or whatever protein. For command line, I think some of you might have already visited, attended Dan's, Graham's course. So that's, you should be aware of it. So you can access directly to that command line from Mac. So if you missed that class, I think it's being recorded, right?
Yes,
and it's on the internet. I'm not sure.
TSL training website.
Okay, yeah. It's on the website, TSL training website. So it's recorded. Yeah. Okay, I will not go into details there. Okay, so once you're familiar with the command line, this is what you are opening into. It's called eBiotools. Good people at eBioinformatics have gathered a lot of programs into one easy to install package called eBiotools, and that is only available for Mac. So once you install this eBiotools on your computer, you can do anything except managing the small reads files. For that, you will need a little bit more advanced installation procedures, or you should use a cluster in this case. And anyhow, for your desktop or laptop, I strongly recommend that you study how to use this eBiotools. That's essential for bioinformatics. So you can do BLAST, you can do to find out what your protein's PI is, and so on. You can do multiple sequence alignments and everything. And eBiotools also has a graphical user interface called eBioX. So when you install eBiotools, or when you get Neil to install eBiotools, let him also install eBioX. So it should automatically install it, but you should check it's been installed. And you can use it through a graphical user interface. Okay, so these are sample programs that comes with eBiotools. So like, for example, cluster W to alignments, Philip to make trees, and then EMBOSS suite. So EMBOSS suite has got like over 150 programs on command line. I think I can show you later on what they are. And also Hammer, which is HMM model search or build, and so on. And then more alignments programs. And then Primer 3, which you can use to design your primers if you have a sequence. Also Muscle, which is another algorithm for doing multiple sequence alignments. And even Staden comes with eBiotools. And there are many others that I don't even know about. A lot more. Of course, yeah, that means that you require to know FASTA format and command line usage. So EMBOSS is also part of eBiotools that can do a lot of things. So usually I use EMBOSS to do sequence alignments, and then you can do database searching with sequence patterns and protein motifs finding, and also codon usage that I use a lot. And then you can look at what the protein properties are. If you have a protein, you need to predict what the PIs and what the contents of that whole amino acid sequence is, and so on. Okay, another thing that's very useful that comes with eBiotools is BLAST. And you might want to say, why bother having BLAST on your computer? You can use, website to do your BLAST. The reason is, if you have the BLAST installed on your computer, you can do your own database searching. You don't have to rely on people having the database that you want to search. So you can create your own database to search. And another thing is, you don't need to connect to the internet. For example, if you're on a plane, if you feel like doing some bioinformatics, then you can do BLAST on the plane. Then you can probably discover some interesting things. And then, of course, you can use like hundreds of thousands of sequences in one go. You don't have to submit one sequence at a time. Yeah, that's it. And also, you can mine the BLAST output with BBEdit, because BBEdit can open very large files. And you can also do PAL scripts to just extract the information that you need. I'll run an example later on. And another thing that I would suggest, but this is purely optional, is to become familiar with PAL programming language, which is very easy to learn. You can learn it in like a couple of months, and then you'll be able to understand scripts, and then you'll be able to use them. Not necessarily write them, but you'll be able to use them and modify them according to your needs. What it can do is it can look through a very large text file and extract just pieces of information that you need, and then report it back and write it to another text file. That's what I use the most, but some people also use as a glue, which can gather all the modules from different programming languages, and then you can tie them together with PAL, especially for websites. So when you make websites. So you can use Perl to create that kind of things. And even more, what better is built in with macOS 10. Okay, I think that's it for the introduction.